Hello. We are now with another topic, that is how to access the protein database. Protein is a macromolecules, and the database of these very proteins are increasing due to dedicated involvement of so many scientists, researchers throughout the world. So when you talk about any proteins, it comes in our mind that this protein has of many properties. For examples, the proteins which has got the multifaceted values. It has the, of course, the sequence of amino acids written from N terminal to C terminals. This sequence achieve the secondary structure, primary structures, and quaternary structures. This sequence, by foldings, they expose the domains or motif for some functions. This protein is also modified to achieve the functions, to achieve the functions. Also, this protein, when it is unfolded or misfolded, then to prevent the harmful effect of this unfolded and misfolded proteins, they undergo the ubiquitinizations, ubiquitinizations for degradations. And of course, the protein synthesis is targeted to different locus. So this data of the protein is increasing day by day. So how this voluminous or the enormous data, they can be made available for the users or the other workers in order to analyze the any proteins. So from lower taxonomical range, for example, the archaea or from the virus to prokaryote and then to all eukaryote, tremendous data are increasing day by day. And this data are managed, organized, and it is assisted by so many bioinformatics tools in order to make its availability to the users. So in this video, I will be talking about the how to access the protein informations resources. So with a simple presentations, I will try to explain all the facts regarding the protein informations resources. So this resources is nothing but it is an integrated publicly available database which can be accessible to all users, which is accessible to all users And this database supports genomics, proteomics, and scientific discovery. So, whatever the proteins are explored regularly, they are discovered and their database are submitted to the PIR. Of course, it was established in 1984 by the National Biomedical Research Foundations in the USA, and all the protein databases are highly annotated, and it has also the analytical tools for the researchers. So basically, if you talk about the PIR is a big organizations where many other database like your Uniprot, Universal Protein Database, or swissprot, SIB, EMBL. So whatever the EMBL, whatever the database are present in this one, they are managed by the big domain, that is the PIR. So PIR, in nutshell, it is a protein sequence database which is annotated, and it covers entire taxonomic range, and within this range about 283,000 sequence database are deposited. If you talk about the features, then these features are: it is comprehensive, non-redundant, that is not repeated, annotated, and it covers the prokaryotes, eukaryotes, viruses, and the archaebacteria. Above that, it is well organized, and all the entries are classified into protein family or the protein superfamily. If we talk about the protein family, the family means the different molecules which has got the related functions, and its gene has originated from the common ancestors. For example, if we talk about the histone gene, if we talk about the histone gene, then there are so many type of the histone genes are there, like H1, H2A, H2B, H3, H4. So all these genes are related in the sense that they have got the common ancestors. So definitely the proteins, this H1 protein, H2A protein, H2B protein, and all these proteins, they together they will form say protein family. So family means it is the relationship, that is evolutionary relationship with the same functions. The protein sequence database, this also include the cross references to other sequences in order to avoid the redundancy. It has got also the structural, genomic, and citations database, along with the publicly available nucleic acid sequence database. So from here you can also move to the nucleic acid database. The database is updated weekly, and full release is published quarterly. So cross references are usual process to avoid the redundancy. Beside this, it has got the additional database like PhD patch X, which include the sequence which are not yet present in the PIR. Then pro class, that is protein data of superfamily, profam protein family data, And PIR databases, I told you that it is organized. So database are structured at three levels. It is a superfamily or family similarity, where the full length similarity is there. Then it has also structured according to the homology domains, domains that provide the local functions, and it is a structured and motive also for structural and the functional sides. So this protein purpose is that this continues to enhance its database to three major database, that is protein sequence database, then non-redundant references sequence database, and integrated protein classifications, where the proteins are classified into superfamily and the family. Here, in order to avoid the redundancy or the repetitions, then NRI, NERF sequence database are there, and the protein sequence databases are there. So this PIR, it has got the two tools. One is it has got the database, and also it has got the tools to retrieve or to mine the protein data from this website. So this allows the access of PIR database, and the database can be accessed by means of the Boolean operators, where the keywords, where any, if you got the search box, so you can type any protein, insulin, or alpha insulin diet. So these keywords include the and or not. So this allow the easy access to the any search. And beside that, it has also got the blast facilities, FASTA facilities, pairwise, pattern match, peptide match, alignment, multiple alignment, like that. So many things are available in the PIR. So one by one, when we will open the website, then I can tell you how to access this PIR. So this is the old home page, where the resources are seen here. If you open the home page, you can go to the integrated protein, TNet, PRO protein oncology, IPRO link, IPRO class, PIRSA, PIR PhD, Uniprot. Like so many links are there. So PIR, it has got the so many type of the resources. So when you have the knowledge of the PIR, so in nutshell, what you can perceive, you can perceive that the PIR is meant for the database, where all the databases are present. If you talk about a references database, sequence database, structural database, all these databases are present. And also it has got the another facilities to retrieve the data. So how to retrieve the data? So data can be searched through the text match. You can write the text, you can search it, or you can enter the ID, then you can have the retrieval. And also the some of the index, PIR DBI or the ORIC DBI. And this database, I told you that the PIR is a big or the large organizations, which includes or which is integrated with the IPOC, IPRO class, that is integrated protein classifications, then Uniprot universal protein database, Uniprot archive, Uniprot knowledge base, Uniprot references, and protein information resource. So this is just a flow diagram which can tell you that what are the things available in the PIR. So in nutshells, we can say that the database of PIR is Uniprot, IPRoclass, PIRSF, PIR PSD. So what I shown you in the flow diagram, this one. So how to access the PIR and what information you can get. So these are the two URL or the website address. So we here we can just open one, that is the protein information resource org. So for this very purpose, it is suggested that you just open one another Google search and you write a simple PIR. So here it has come protein information resource. You click this one and you can get the web address. So this first one is the welcome to PIR. This is the web address. You click this one and you can get the home page of the protein information resource. So this is the home page. Okay. So I told you that the home page has got the protein oncology, then integrated protein, translated modification resources, then literature information and knowledge link. It has also have the text search, peptide search. These are the tools. So what are the resources they are available? If you see about PIR, you can get the history, publication, staff, all these things. If you get the resources, then the PTMNet, Protein Oncology, ProLink, ProClass, PIR, these are the all the resources are there. If you have the download, then you can have the file transfer protocol center, then PIR SF is there, IPRO class is there. So basically, if you want to know some of the analysis of the any proteins you have retrieved, any sequences, so this PIR that give you the facilities to text search. If you have protein informations in the form of the writings or in the form of the literatures, you can text search, or you can have the batch retrieval. Then you can also the peptide match, which peptides are matching. Then you can have the also the facilities for the pairwise alignment. If you just have one pair, one protein sequences, you want to align with the another protein sequences, so how much degree they are aligning together. So alignment that shows you the homologging. If you have got the multiple alignment, I told you about in the cluster W, where in the cluster W multiple sequences are aligned, and that tell you about the conserved domains, then the where the substitutions are there, where the deletions are there, all these things are. And another one new thing is that from this very PIR, you can also have the knowledge about the how to calculate the compositions of the amino acids, that is how much frequencies are there, and also the molecular weight of the total polypeptide. So one thing that is the you have already know that multiple alignment how it can be done in the cluster W, uh, and the text search. First, let us see the text search. So in the text search, when you will just open it, so you get an page, page where the select a database, select a field, or insert query below. So we are just all fields are there. So in this we have to filter it. So we have filtered it by the gene bank account or the gene name, or you can have the also the protein. So I am just here writing the protein name. Okay, this is the, or you can also have the Uniprot knowledge base ID. So protein name, this is the one of the easy task. So when you will given an any assignment that search the protein information, search the any protein information from PIR. So here you can just select a database IPRO class or PIR. Here I am selecting IPRO class. You can also select the PIRSF. So let's move back. Hmm. Okay. So in the text search, You write the protein name. So here the protein name it has come, and write any names. For example, I have written the caspase. Okay, so caspase. So caspase in enzymes which are involved in the apoptosis for just degrading the all the materials of the dying cells. Now add input box, then search. So here it has come the informations of different type of the proteins. So when you have got the IPO class, so here also the PIRF SID. So you can also you can copy it or you can directly open it. When you will open it, this one, then also you can get the informations. So PIRF entry I have kept. Then here you have not PIRS number is there, PIRSF name is there, PIRF size is there. So total entries 50. So here you can also have the other informations like uh, which cysteine type endopeptide activities their cysteines are involved in the peptide. Then another cysteine is the protein containing complex binding protein. It is involved with the apoptotic process. Also, it is involved with the regulation of the apoptotic process. So many informations and structures you can get from this text search. So let us come to the our this. You can go back, and another facet is that it provides to the user is that you can know the molecular weight. So in order to search the molecular weight of the any proteins, so you click this search tool, molecular weight and compositions, because this will allow the retrieval of the sequence molecular weight. So here the two options are here. Either you just enter the Uniprot knowledge base identifiers, and if you put knowledge base identifiers, then if you are writing or if you are pasting only one, then no need to just have the space. But if you are pasting two, then you will have to, okay, let me see Uniprot identifiers. Okay, so this is the ID. Okay, this is the ID IMSD human. Okay, and this is the, so this you can copy it and you can paste it. This also, this is the accession number. So if you are having the two unit plot ID or three, then you will have to keep the space. But the simple thing is that you can load the sequence. You can load the sequence. So for any sequences, you will have to go back to have the sequencer. So for that purpose, you have to open the again the NCBI, which is the step, initial step in order to download or copy any database. So NCBI, okay, and from this NCBI, you just from the home page, you filter the proteins, you type the name of the proteins, then go for the search, you can get the data. So for the this proteins, uh, this protein is there. Again, I'm talking about the caspase enzymes. Okay, caspase enzyme search. Here also you can have the a filter of the human or the Drosophila or the C. elegans. So from this way from also you can filter. But I'm not going to the filter options. Either I'm just selecting the first of the sequence. So this sequence is death related CED genes, caspase isoform G from isolated from the Drosophila. And here the bioproject, nucleotide, PubMed, taxonomy, gene peptide, identical proteins, all these things are given. So we need a FASTA sequence. So you just click this one. So then yeah, the FASTA sequence of this very protein will come on the page. So it is the FASTA sequence. You just select the sequence by this one command, you select the sequence, you copy it, and directly you just enter here. You just paste it and have the search. So once the search option is clicked, then the our tools will create our tools will just give you the composition of the amino acids. Here the total amino acids is 502, and all these amino acids, there are 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 21 22. So there are 22 of amino acids, and this is also included X, this is the unknown one. 23 amino acids are here, and this 23 amino acids, it has got the different frequencies. For example, the alanine is present. So alanine is how much is present. So it if you just calculate its percentage in the frequency, then what you will see, you will count the number of the alanine here, and if divide by the 503, and you multiply with the 100, so you can get one. Okay, so 7.6, so it may be having, you just count this one. For example, say in this sequence, in this one, no alanine is here. Okay, so this has been already calculated. So number of, because amino acid count is 38, number of alanine is 38. Okay, number of cysteine is 10. So which amino acids are very less in number? This is W. So W, I mean, you can have just search it what the W indicates. W might be in. Let me see here. What is W? Okay, you can see. And in the this one, this is the formula how to calculate the molecular weight. So molecular weight is calculated because average molecular weight is 110. Okay, average molecular weight is 110 for any amino acids. Okay, let me see you. This is the average amino acids. It is one. Sorry, it is 110. Okay, but here it is given alanine. It has got the molecular weight of 89.01. This so what you want to do, you just add. You add all these. Okay, when you will add, then whatever the value it will come, let it be the x. Okay, now this all are interlinked by peptide bond. So this is the sum of the individual residues. I have written it is the x. Okay, so what is the molecular weight? Molecular weight is of water is 18.015, and this is the number of residues. So how many number of residues are? 502. This is the 502 minus 1, because if you why this is minus 1, because if you say 4 amino acids, okay, this is the AA, this is AA, this is AA, this is AA, this is 1, this is 2, this is 3, this is 4, and here is the peptide bond 1 2 3. Okay, 1 to 3 peptide bond. If you see 4 5, where A1, AA2, AA3, AA4, AA5. So, and here is the peptide bond 1 2 3 4. Okay, so it means that key the number of amino acids is 4, but the peptide bond is 4 minus 1 3. So here also, if the number of residues are 502, then you just minus it, you can get the result. So this molecular weight has been calculated by using this very formula. So I think that this is a very easy task. If you want to know that which protein has got the larger size. Suppose that you have been given caspase, you have given the insulin, you have been given the some of the trypsin like that. So you'll have to search which has got the more molecular weight, more molecular mass, which protein has got the more molecular mass. So you can calculate in the kilodalton or you can calculate in the Dalton. Very much you know that 1 kilo Dalton is equal to 1000 Dalton. So this is the Dalton. Molecular mass is a Dalton. So if you will remember it, you can divide it by 1000, 1000, then you can get the 56.863 kilo Dalton. So this molecular weight is 56.863 kilo Dalton into 100 like that. 1000 so it is the 56083. Okay, I am not considering here this one. So I think that this is a very easy database or the website putting information source where you have to perform any type of the task that has been given to you. Hope you can understand it too. Well, if any queries are there, you just write on my website. That is the one website is there. I am writing here website, and that website is djane1996 djane djane1996 dot word press dot com. Okay, or you can just give any information in the google class google classroom, so we can solve your queries. Thank you very much.